Hi, my name is Kevin, and we're going to look into the effect of prolonged valine deprivation on mTOR activation. My mentor is John Blennis at John Blennis Lab, Pharmacology at Weill Cornell Medicine. Some key words are mTOR, which is mammalian target of rapamycin. It's a major regulator of growth in animals and controls most anabolic and catabolic processes in response to nutrients and nutrient-induced signals. The next key word is valine, which is one of the essential branched amino acids and must be taken in by food since your body can't produce it on its own. Then there's PI3K/AKT, which is a pathway involved in cell growth and proliferation and is activated by growth factors like insulin. And finally, there's RAG-GTPase, which is a regulator on the lysosome involved in cell growth and is activated by amino acids. Alright, so now let's look at the big picture. mTOR is a regulatory protein and is divided into mTOR complex 1 and mTOR complex 2. mTOR regulates many cellular processes like gene expression, protein synthesis, and cell division. mTOR can be activated through growth factors like insulin, shown here in the yellow dot, and that goes through PI3K/AKT and activates mTOR complex 1. mTOR can also be activated through amino acids, so that's shown in this gray dot through RAG-GTPase. And finally, PI3K also activates mTOR complex 2, which in results reactivates PI3K. And overactivation of mTOR is seen in over 70% of cancers. Thus, understanding the regulatory mechanisms of mTOR may be vital in the treatment of cancers. So now let's look into my hypothesis. Since many amino acids play a role in mTOR regulation, I want to see the effect of prolonged valine deprivation on mTOR activation. I hypothesize that prolonged valine deprivation will cause a decrease, so an initial decrease in mTOR activation before reactivation of mTOR. So now let's see the methods in which I'll do this experiment. So first, mouse embryonic fibroblasts will be introduced into solution of DMEM supplemented with fetal bovine serum and other nutrients. One sample will be allowed to develop under ideal conditions, so that's this one with the gray dots representing the valine. And then the other will be the absence of valine, so it doesn't have the gray dots. And then cells will be collected at certain time intervals from these solutions, and like metabolite profiling will be done to determine the mTOR activation. You can see here with the graphs and stuff. So what are the expected results? With valine, we should see constant mTOR activation since it's under ideal conditions. However, without valine, we should initially see a decrease in mTOR activation since RAG-GTPase doesn't have the amino acids required for it to activate mTOR. However, over time, we should see a reactivation of mTOR since the lack of amino acids would cause a activation of PI3K, which will eventually reactivate mTOR. If these are the shown results, then more research should be looked into how the absence of amino acids activates PI3K, which then reactivates mTOR. And now finally, what is the significance of this data? This data might be significant in treating cancer since many novel cancer treatments involve depleting cancer cells of essential amino acids like valine. The fact that mTOR is reactivated under valine deprivation may add another level of complexity that scientists need to consider before employing drugs that restrict amino acid absorption.